biology, and he will tell us about resolution of regulatory conflicts through genome 3D reconstruction. Mike, take it away.
Cool. Hi everyone. Thanks for the kind introduction. It's a great pleasure to be given a chance to follow Luca and continue talking about 3D genome organization and how it can control gene expression. And what I want to focus on today is to talk to you about how we can use this information to try to understand the impact of genomic rearrangements in the genome and how this relates to disease. So to start with, I want to just do some upfront acknowledgements. So a lot of this work was done during my postdoc in Stefan Munlos's lab by the work together with two of my very talented PhD students, Alessa Ringel and Konrad Hützig, who are currently finishing up their PhDs and looking for postdocs. So I'd strongly encourage you to get in touch if you'd be interested. And it was also supported by our master students, Patricia and Janina, as well as a number of friends and collaborators around the world in the Cavalli, Nicodemi, Meissner, Kin, Solivay, Hageling, and Vingron labs, and couldn't have been possible without the MPI FACS facilities and mouse facilities and funding, and also going forward my new lab. So, also I wouldn't be a new junior group leader if I wasn't going to do some shameless self-promotion. We are recruiting for PhDs and postdocs currently, so again, get in touch. So the question following up from Luca that I want to really focus on now is one that's fascinated me for many years, which is how genes are precisely expressed in time and space, because we know our complement of 20,000 protein coding genes need to be carefully controlled. And this drives cell identity, it drives embryonic development, and it maintains tissue homeostasis. And so modifications or disruptions to gene regulation or gene transcriptional regulation contribute to both evolution but also human diseases. And these can range from congenital malformations to cancer as well as autoimmune diseases. So for myself, I have a particular interest in the autoimmune diseases because in the final year of my PhD, actually in nearby Edinburgh, I acquired alopecia, which is likely contributed to by gene misexpression, which causes my T cells to attack hair follicles and cause them to fall out, removing this sort of glorious insulating layer of hair, which I'm sure you can imagine. It's not normally this warm in Scotland. So it's a different experience with no hair. But the good news for patients like myself, but also for people studying gene regulation, is that we can now map these disrupted gene expression states pretty well using new technologies like single-cell RNA sequencing, which I'm sure you all heard about during this conference. And these can be used to better design therapies. But one of the big major challenges is still that our ability to now map these disrupted gene expression states now far exceeds our understanding of how they're encoded genetically. And until we can understand this, it's very challenging to reliably predict or treat the disruption of gene expression in disease. And that's the problem my lab and many of you here probably want to help solve. So what do we know about transcriptional regulation? Well, what we know, like Luca very, very elegantly introduced for us, is that the instructions to tell a promoter where and when to transcribe a gene are not found in the gene itself. Instead, a lot of this information is distributed in the vast 98% of non-coding DNA that surrounds genes. Specifically, a lot of the information is in these short sequence elements that we call enhancers that each have a defined pattern of activity. And these activities then get transmitted to target genes, often bypassing hundreds of kilobases or even megabases as they do so. This is incredibly long-range communication. Now, the activities of enhancers and promoters is governed, as Luca said, by the binding of transcription factors and chromatin modifiers. And these then alter other features, such as histomarks, chromatin accessibility, or DNA methylation. And we can actually use these this unique biochemical signature and genomics to actually identify these putative elements and sort of define their activities in different cell types and tissues. So we're still understanding how the actual enhancer transmits this activity to the target gene, as Luca suggested. But importantly, beyond regulation by the epigenome, we also know that the 3D structure of the genome itself is a critical regulator of enhancer function. And that's because enhancers seem to activate their gene targets by moving into their promoter's close physical proximity in 3D space through chromatin folding. And so the 3D structure of the genome is really an essential layer of gene regulation that we need to study. And as Luca has, again, mentioned, we can analyze these 3D structures using many different technologies. One of the predominant ones we use is these chromosome conformation capture approaches like Hi-C. And just to reiterate, this is the same maps that Luca has showed before. We show this data in heat maps where where the pixel represents, and the intensity of color in the pixel represents the frequency of contact between the two DNA segments at the end of the diagonal. And so in this way, we can really build up a genome-wide all versus all picture of chromatin structure. And when we do this, we see that the genome has incredibly complex levels of organization at multiple different scales. And one of the most studied of which, and is involved in diseases but most characterized, is topologically associated domains, or TADs. And these represent preferentially self-associating regions of the genome that are separated from one another by these insulating boundary elements. And they represented in the Hi-C maps as these triangles. And what TADs seem to do is to allow enhancers to physically contact and activate all promoters found in the same domain, but not in neighboring regions. And in this way, TADs are really restricting the regulatory information that any given gene is being exposed to. And these have been demonstrated by many different experiments, including the one that Luca described. And as a result of this, the relevance for human genetics is that then structural rearrangement mutations like large deletions, duplications, translocations, and inversions can create novel combinations of enhancers and promoters within shuffled TADs that can drive gene misexpression or disease. So in this case, gene A, for example, has lost access to its purple enhancer, and gene B has now ectopically acquired it. And as a result, gene A loses expression in the hindbrain, and gene B ectopically gains it. And this misexpression can drive disease. And there are a number of different examples of this, but for example, the misexpression of Indian hedgehog in the developing limb, caused by it being placed into an inappropriate TAD with inappropriate enhancers, causes the congenital malformation polydactyly, where you get too many digits on the hand. So clearly, alterations to TADs can create these pathogenic regulatory conflicts in the genome that cause disease. And so together, TADs, enhancers, and promoters provide us with a really useful framework for understanding how regulatory information is both encoded in the genome, but also how it can be partitioned. And importantly, this has been used pretty increasingly in human genetics to understand the causes of genetic disease. So we know, for example, that TAD-disrupting mutations that create regulatory conflicts, like the one I just described, can contribute to a number of cancers, as well as limb malformations or congenital malformations. We also know that targeted deletions that affect specific enhancers themselves, alter the dosage of specific enhancers, can contribute to leukemia or craniosynostosis, and even sequence variants, point sequence variants within enhancers can alter their activity by altering transcription factor binding sites, and this can contribute to, for example, craniofacial abnormalities, but also very likely make way more common and more prevalent disorders such as autoimmunity or diabetes, because we know that the enhancers associated with these processes are highly enriched in disease-associated GWAS variants. So clearly, disruptions to the regulatory genome are a major cause or source of disease. So with that, the way I've sort of described it, it seems like, well, we should be able to therefore use this framework to predict how any given genomic rearrangement or mutation is going to impact gene regulation. However, as sort of everyone in this audience knows, that really isn't the case. And it's still very, very difficult to predict which non-coding variants and rearrangements are going to be benign or pathogenic. And despite the fact that most of the variation in the human genome comes in the form of these non-coding variants and rearrangements. And I think that likely contributes to why we can still roughly only solve roughly 50% of these rare genetic syndromes, for example. And in particular, what we often see in the genome is that mutations that should seemingly be pathogenic because they create novel combinations of enhancers and promoters within these TADs are actually benign. They do not drive gene misexpression or disease. And this suggests that there must be additional mechanisms in place within even these rearranged structures that still restrict enhancers to their correct gene targets so you don't get disease. And these are the mechanisms that we wanted to identify in the study I'm going to tell you about now. So we wanted to identify what the mechanisms were that resolve regulatory conflicts in the rearranged genome in rearranged TADs. And I think this is quite informative for how the components of gene regulation work. But we didn't do this by looking in patients. Instead, we looked at another example where rearrangements and conflicts inevitably arise and need to be overcome, which is in evolution, obviously. And specifically, we examined the evolution or the emergence of a regulatory conflict in the evolution of a specific TAD that contains the FAT1 gene, as well as all the enhancers required for its activity. Now, if we look at what activity this TAD drives for the FAT1 gene, we see this beautiful pattern of expression that we can see with whole mount in situ hybridization, where the color, the purple color represents the level of mRNA. And we can see FAT1 has this beautiful pattern of expression in the face, the eye, the developing ear, the mammary glands that you may not be able to see here, as well as the developing limb. And if we focus on this limb expression, we can see it's been conserved across 300 million years of evolution. So a similar pattern of expression is found in the limbs of opossum embryos, so a marsupial, and even in the chicken, so a non-avian dinosaur, because non-avian dinosaur sounds cooler than bird. But we wanted to then, within this, see if we could identify if what regulatory conflicts arose at this locus over evolution. So we performed high-C to map TADs and ATAC-seq to map putative enhancer elements. And if we look in, and we did this in the limbs of each of these species. So if we look in the chicken at the real data now, you can see this large red triangle, which is this region of preferential self-interaction that is the FAT1 TAD here, right-hand side of which you have FAT1. And ATAC-seq identifies putative enhancers throughout the whole domain. Okay, and then if we go into the opossum, a non-placental mammal, we see a very similar TAD structure, and again, enhancers distributed throughout. Interestingly, there's a synteny break in all marsupials on the left-hand side of this domain that brings in an entirely different part of the genome to be neighboring this domain, but importantly, the TAD remains as an intact unit, so it seems to be a functional unit of the genome which is being maintained. And if we now go into placental mammals like the mouse or also human, we see again a very similar TAD structure with enhancers distributed throughout. So far so good. This seems to be following the rules that we understand for gene regulation. However, there was one major regulatory conflict that emerged in the evolution of this, in the evolution of placental mammals, which is that a new gene, ZFP42, inserted into the center of this domain. Now, based on what I've told you, you guys should now be able to predict how this new gene is going to be regulated. It should be exposed to the same enhancers as FAT1 and therefore have a largely overlapping pattern of activity. However, what was sort of really surprising to us, or frustrating perhaps, is that this is actually not the case at all. Instead, ZFP42 has an entirely independent pattern of regulation, and in fact, in this stage of embryos, it's entirely not active whatsoever. And so like in patients, we see it doesn't seem to follow the rules of how we think TADs and gene regulation work. And so what Alessa and I wanted to do was to understand, well, how can ZFP42, what mechanisms allow ZFP42 to be regulated entirely independently of this gene FAT1? So what mechanisms resolve the regulatory conflict that evolution created? And to address this, we took a strategy which was pretty comprehensive, where we examined two different situations in the mouse where ZFP42 and FAT1 are differentially regulated, in the limb of the embryo, where FAT1 is expressed without ZFP42, and in ES cells, where both genes are active together simultaneously. And we took a strategy where we would map the chromatin structure and the epigenetic state of the locus using a combination of genomics approaches like ChIP-seq or Hi-C. And also, we would supplement this with directly visualizing the structure using super-resolution microscopy as well as polymer modeling. And then once we'd mapped these states and structures, we can now, with new CRISPR technologies, really dissect their function by disrupting them. And then we can actually also take these mutants into mice and actually generate mutant embryos and live mice from these that we can study. And so if we first look at the limb situation, which again is where FAT1 is expressed without ZFP42, I can now walk you through how we saw the mechanisms that resolve this regulatory conflict. So just to remind you all now, ZFP42 and FAT1 in this case are located within the same large 3 megabase size TAD, so it's a really huge domain. And we can actually map putative and active enhancers and promoters within this domain using H3K27 acetylation ChIP-seq. And you can see there's a cluster of enhancers on the left-hand side that here from the peaks in K27 acetylation that are active in the limb. And these are physically contacting both ZFP42 and FAT1, but only FAT1 is being activated by them. So we wanted to see what mechanisms could exist to allow ZFP42 to evade these activities. So we came up with 3 different hypotheses that we progressively, during Alessa's PhD, disproved, much to her sort of frustration. And I'm going to walk you through them. So the first one was that, well, maybe unlike sort of the argument that Luca was going before, that all information is sort of freely shared between within a TAD, maybe ZFP42 just doesn't actually receive enhancer activities. Maybe it's its position which is the problem. And so to test this, we took advantage of a Laxi reporter gene, whose expression is driven by a minimal promoter that lacks the ability to be transcribed by itself, but it can be activated by enhancers. And so by integrating this at different positions in the genome, you can see which enhancers are available to each gene. And so we wanted to therefore integrate this at different locations in the TAD and see if they could respond to enhancers. So if you integrate this in the vicinity of the FAT1 gene, it's able to be activated by FAT1 enhancers in a FAT1-like pattern of activity. So we see it's expressed in the eye, the ear, the mammary glands, and the developing limb. And this is very similar to the endogenous FAT1 gene itself. But what was really sort of surprising is that this sensor could pick up and be activated by FAT1 enhancers anywhere inside this TAD, even when it's almost 3 megabases away, or when it's in the direct vicinity of the endogenous ZFP42 gene. So this is very similar to the result that Luca Giorgetti was showing earlier. And this actually isn't the case when the sensor is integrated outside of the TAD, because now it can't pick up activities because the TAD boundary is blocking their contact. And so FAT1 enhancer activities are actually easily transmitted to ZFP42's position. So that's not the case here. So instead we thought, well, maybe it's that ZFP42's promoter just isn't compatible with FAT1 enhancers. It doesn't matter if it can contact them, it's not going to be activated by them. So we took a strategy where we replaced the minimal promoter that we've been using before with the promoters of ZFP42 or FAT1. And then we integrated these again back with the Laxi reporter, 20 kb from the endogenous ZFP42 gene. Now, perhaps unsurprisingly, FAT1's promoter can be activated by FAT1 enhancers. It's compatible with them. So it produces a pattern of activity in the embryo, which is very similar to that of the previous reporter, as well as the FAT1 gene itself. But what was really surprising is that ZFP42's promoter could also be activated by these enhancers when it's repositioned only 20,000 base pairs away from its endogenous location. So it seems that ZFP42's promoter is totally compatible with FAT1 enhancers. And I do just want to say, though, that we do see some variability in the responsiveness of the promoters to enhancers. So we do see, for example, FAT1's promoter, which maybe you can't see here so well, but it gains activity both in the forebrain as well as in the apical ectodermal ridge, which is the edge of the developing limb. And this isn't detected by the other promoters. So there is some specificity that exists, but it cannot explain why the endogenous ZFP42 gene is off. So at this point then, we have the same promoter can be inactive at its endogenous position, but activated ectopically by enhancers when it's moved 20 kb away. And so the only mechanism we could think of at this point is, well, there must be context-dependent silencing at this endogenous location, which keeps the endogenous ZFP42 promoter off. And so we looked around for silencing mechanisms for a while, and we settled in the end on DNA methylation. Which is a known repressive feature that we can detect with whole genome bisulfite sequencing. And we can see that in ES cells, when ZFP42 is active, its promoter is poorly methylated, but it becomes heavily methylated in the limb once it's inactive. And this is very different to FAT1, which is unmethylated and active in both tissues at its promoter. Okay, so what we obviously want to do is eliminate this methylation at ZFP and see if we can sensitize it to FAT1 enhancers. And so we did this by eliminating the de novo DNA methyltransferase, DNMT3B, which is partly responsible for laying down methylation in the embryo during embryogenesis. And we see that we can eliminate a fraction of the methylation the promoter normally gains. Not everything, but some. And now we want to see, does ZFP get turned on when you get rid of the methylation? And you can guess the answer, perhaps. The answer is yes. ZFP gets activated by 6-fold in DNMT3B knockout limbs. And if we look at the pattern of the distribution of this activity, we see it's activated in a very similar pattern to that of the FAT1 gene, as well as the sensor that we previously relocated. So it gets turned on in the mandibles as well as in the developing limb. So demethylation is sensitizing ZFP42's promoter to these enhancers. And what I think is really interesting is that when this happens, FAT1 expression goes down by 50%. So the misregulation of one gene causes seemingly the misregulation of another. So it seems to have created a regulatory conflict, which I think is interesting. So to summarize in this tissue, we see that enhancers can indeed activate promoters promiscuously throughout their TADs, but context-dependent repression can prevent specific genes from responding to them. So that explains the limb situation, but what about in ES cells, which is where ZFP42 and FAT1 are now active together? So to look at this, we repeated our high-C analysis in embryonic stem cells, and what was really surprising, which I think you can probably already appreciate, is that despite the fact that TAD has been conserved for 300 million years of evolution in the limb, it's just massively reconfigured in these ES cells. Suddenly, we see that ZFP42 and FAT1 disengage their interactions with these deserts 1 and 2, and you can see this on the white lines in the high-C map, and here and here. And simultaneously, deserts 1 and 2 lose their interactions with each other, so you can see this square in the center of the TAD, which has lost contact. And so now the TAD has been partitioned into 4 discrete domains of D1, ZFP42, D2, and FAT1. And we can actually directly visualize this using super-resolution microscopy. So we can see that the ZFP42 region shown in green here, when inactive in the limb, is closely intermingled with these deserts 1 and 2, and it's closely attached to the nuclear envelope, which is shown in red, which I'm going to talk about more in a few minutes. Whereas ZFP40, when it's active in the ES cells, ZFP moves away from the nuclear envelope and is now poorly intermingled with deserts 1 and 2, which now also exist as separate structures. And we can also further visualize this using polymer modeling, which is based on it's computational modeling based on our genomics data. And so now you can see the ZFP region shown in orange here, when inactive is really just, you can't even barely see it because it's buried by these gene deserts at the nuclear envelope, whereas when it's active, the orange region is escaping and separate from the blue regions. So TAD partitioning, in this case, is isolating FAT1 and ZFP into separate, smaller domains. Now we can look at how that restructuring affects the regulation of the locus by mapping active enhancers and how that activity was redistributed using H3K27 acetylation ChIP-seq. So now you can see the limb enhancers are inactive, and instead enhancer activities have been redistributed to clusters that are found within the little isolated domains of either ZFP42 or FAT1 itself. And we can actually show using deletion experiments that these independent clusters of enhancers drive the independent activity of either ZFP42 or FAT1. And this is so much so we can actually totally delete ZFP42 and all of its enhancers, and FAT1 is totally unaffected. So the restructuring in this case has just facilitated totally independent activity of the 2 genes by controlling which enhancers each of them are exposed to. So clearly then, TADs can have flexible structures, which has been a really underappreciated feature, that are highly tissue-specific, and these can restrict genes with different enhancers in different settings. And this resolved the regulatory conflict in ES cells. So to summarize then, what we can see is that evolution resolved the regulatory conflict that was created by ZFP42's insertion through 2 highly tissue-specific mechanisms. On the one hand, ZFP42 can evade the enhancer activities it's exposed to through context-dependent silencing. And on the other hand, restructuring in 3D space can just isolate genes with different enhancers that control their activity very distinctly. And this actually allows a single locus to easily incorporate multiple entirely independently regulated genes without any conflict. It's totally fine. And importantly, which I think is the biggest take-home message, is that we see this ability to resolve regulatory conflicts is a generalizable feature of the genome. Most TADs in the genome contain multiple independently regulated genes with totally different patterns of expression. And so mechanisms like these seem to be operating all over the place all of the time. And I think this explains likely why many variants and rearrangements that seemingly should be pathogenic are actually benign. It's because additional mechanisms beyond TADs are restricting enhancers to their correct gene targets, and that likely limits the pathogenicity of many rearrangements. And importantly then, by incorporating these data from these additional mechanisms into our sort of variant interpretation, I think you can improve the efficacy of variant interpretation. Yes, so that was quite satisfying that we could solve the case of this specific locus, but I think it raises obviously a more generalizable problem, which is that the reason deciphering transcriptional regulation, the reason why this is so hard, is because it's encoded in the interplay between enhancers, promoters, and TADs, but also between epigenetic regulators, of which there are many. I've only mentioned a few of them here, and the fact that the genome's 3D structure can change radically between different cell types. And there are almost certainly missing regulatory features that we really need to start incorporating. And importantly, these components can be assembled in many different combinations at different loci, and therefore give genes very different forms of regulation. And that creates three major challenges to be able to determine how any given gene is regulated and how it could be dysregulated. So what we need to be able to do is, well, we know that each of these features can be very different in different cell types. So we need to be able to measure these chromatin features in highly complex tissue environments across many different cell types to collect this data. We need to be able to, we know that all of these features act together to function, and therefore we need to understand how they interact and influence one another to control gene expression as a unit. So we need to measure multiple things at the same time. And we also need to be able to identify and dissect the function of these missing features at genome-wide scale, so we don't just go through this locus-by-locus dissection, which takes a PhD student 5 years and a lot of frustration. So how can we do this? Well, I think one of the most powerful technologies and ways out of this problem is to use single-cell multiomics, which can use sequencing to map multiple features together in the same cell, such as A and B. And by applying these to, for example, tissues, we can then map in single cells and cell types these different epigenetic profiles, so we can solve problem one. We can also apply these to situations of perturbation, like in disease, where one feature like A is broken, and then we can directly read out the consequences for another B in the same cell, and therefore establish a causal relationship and determine how they interact to function. And then obviously we can apply these to look at missing components of gene regulation. And so I want to just tell you very briefly about a new technology that we can use to solve these three problems, which is single-cell DAM&T, which I've been working to implement in mice in collaboration with Job Kin's lab at the Hubrecht Institute in the Netherlands. And this allows us to now map the binding of any protein of interest to chromatin at the same time as transcription in the same cell from any complex tissue in vivo in the mouse, but also other complex tissue settings. And I want to tell you now, I don't really have time to tell you about how the technology functions, but I want to show you an example of how we can use it to solve or understand better a missing component of gene regulation, which in this case is a higher level of organization that exists beyond TADs, which unfortunately is called LADs. I know, a very similar abbreviation. But this is a high level of organization where the genome is organized by activity, where we know where heterochromatin attaches to the nuclear envelope in these repressive lamina-associated domains, LADs, or positions in the nuclear interior when active in non-LADs. And we have many reasons to believe that these LADs contribute to gene regulation, because we know mutations in the nuclear envelope proteins that are implicated in attaching heterochromatin in LADs cause a wide variety of diseases that are collectively called laminopathies, but have we're still understanding their modes of the molecular causes, and these range from Emery-Dreifuss muscular dystrophy to Hutchinson-Gilford progeria syndrome. And so we want to understand the function of LADs, and so we wanted to ask just a very simple question. We know that loci need to transition between these two environments during embryonic development, and we just want to ask the simple question, well, when does LAD heterochromatin get released in order to facilitate genes turning on in development? So we applied DAM&T to map changes to LADs and transcription over time at multiple stages of mouse embryonic limb development, and we also profiled a number of tissues outside the limb as sort of outgroups, and we did this together with the Kind lab. And we can use the transcriptomic component of this technology to resolve cells as they're differentiating over time in this highly complex tissue setting. So I think we're all familiar with UMAPs. Each dot represents a single cell, and the distance between them represents their transcriptional similarity. And so you can see limb skeletal muscle cells are over here, whereas limb mesenchymal cells distribute down here, where and these contribute to the bone, cartilage, and connective tissue in the limb. But if we zoom in on the mesenchyme here, we don't just have to color the cells by cell type, we can also color them by developmental stage of origin. And now you can see here, these are cells from the E14.2 embryo, which is the lateral plate mesoderm, which is where the limb is going to originate from. And then in E95, they differentiate to create a critical cell fate decision. They decided they're going to be naive limb progenitors. They know that they're going to be the limb. And then they differentiate and radiate outwards to form these much more complex cell types in the later E13.5 embryo. Now, importantly, we have measurements for LADs and transcription in the same cell. So now we can ask, when does changes to one happen relative to the other? So we can show that by coloring the cells instead by their degree of laminar association, or mRNA, where red is high, gray is low. And you can see when a critical gene like limb gene like SHOX2 is inactive, it's highly laminar associated in the lateral plate mesoderm. And then the moment you go into these progenitors that have decided that they're going to be the limb, suddenly this laminar association is released. However, this happens way before the genes actually turn on in later cell types. And so we think that this LAD pre-release is serving as a licensing step that allows genes to turn on at later stages of development. But then we don't just have to look at the genes themselves. We can look also at how the entire TAD landscape behaves. And so we say, do entire TADs detach from the nuclear envelope together with these target genes? And so if we look at the SHOX2 TAD, we can see this huge sort of megabase-sized domain, and SHOX2 is located here. And if we create average LAD profiles, so frequency of contact with the nuclear envelope for cells of the lateral plate mesoderm before releases happened, and in naive progenitors after releases occurred, we can see indeed it's not just the SHOX2 gene which detaches, it's the entire TAD domain. And we can even decompose these naive progenitors at different time points to early, intermediate, and late, and we can see that this release happens progressively over time. And now we can actually go also one step further, because single-cell DAM&T can be integrated with other single-cell modalities that measure other features, like chromatin accessibility using single-cell ATAC-seq. And then we can start asking, well, what other features coincide with LAD release? For example, the activity of regulatory elements. So we can ask, and the answer to this question is yes. We see that regulatory elements by ATAC-seq, a number of them that are distributed throughout the TAD that are shown in red boxes here, these gain accessibility in early naive progenitors at the moment where LAD release is occurring. So we think that transcription factors are binding to these sites to modify chromatin in order to drive the LAD release. So with that, we can see the LAD heterochromatin seems to get released at TADs, entire TADs, before genes even turn on, at critical cell fate decision windows, and we think this is a licensing step that facilitates gene activity. And we see that this is linked to the gain of accessibility at specific elements, but is delimited ultimately by TADs. And the goal now is to see if this process goes wrong in these diseases I described earlier, these laminopathies. But it highlights how all of these features have complex interplays with one another that we need to understand gene regulation. So with that, I'm going to start summing up. So what I've hopefully shown you is that transcriptional regulation can indeed be understood if we integrate multiple features together simultaneously. And you can use this information to better explain genomic variants and predict which are pathogenic or benign. That's the idea in patients. And ultimately, though, we need to go away from just doing locus-specific dissections. We need to have genome-wide analyses. And a way of doing that is to implement single-cell multi-omic approaches to start inferring both relationships between features, but also to determine what each feature is actually doing. And that's what my lab is going to be going to in the future. So with that, I'll just finish with the acknowledgement slides and just be happy to take any questions. Thanks.
Thank you, Mike, for this truly educational and inspiring talk. While people please go to the microphones to ask questions, I will actually ask questions because I have many. So in the first part of your talk, you told us about that gene that's now inserted ZFP, but you didn't really tell us why do we and the mouse have it. like what's the function? And then follow-up question, the methylation mechanism you show us, have you tested this hypothesis also in other TADs, whether this is true in other loci?
So in terms of what ZFP42 does, it's interesting. If you have it knocked out, in mice, you move away from a Mendelian ratio for HOMs, so that it seems to be pathogenic, but we don't really understand why. There's ideas it's involved in pluripotency, but for us it seems to be dispensable for that. It's also expressed in the placenta, and it evolved in placental mammals, so it sort of has a tempting thing that it's involved in placental function, but we haven't really looked into that. And then in terms of whether methylation affects other loci, what we've seen is that if we eliminate methylation, other genes are misregulated in the limb. And it's not, and there are genes that lose methylation, some get misregulated, some that don't. The ones that get misregulated are found in TADs that contain other genes and enhancers that are active in the limb. So you're only going to be misregulated if you're located in an environment that contains other enhancers that can turn you on. If you're not in that environment, then you don't care. So methylation actually is, in that tissue, dispensable at many loci because there's no problem. But in other tissues, there would be. Yeah.
Before we go to the room, I have a bit of a follow-up. So the zinc finger proteins are a fast-evolving group of paralogs in the mammalian genomes and are usually as a tug-of-war to control transposition. So basically, it's a bit the similar point that Malte was doing. So is this not a particular group of genes? And so this is why you see this. Can you envisage another model for the other gene? Or you think this is a rule?
Yeah, so like, is ZFP involved in its own methylation, for example? Is that? Or?
Yeah, and the ZFP needs to be protected. So they are parallel. So they move around because they are duplicated. They land somewhere, and they have to be, quote unquote, I'm inventing here, but protected.
I see. Yeah. So what you do see, so ZFP42 is actually a retroviral copy of another gene, YY1, and this creates a unique problem because unlike sort of tandem duplications, which is often how you get other genome amplification events, this one allows you to hop into new environments. So yeah, it might be that this needs to be inherently able to behave independently from its surroundings in order to, But we never looked at whether gene hopping events display more of this methylation regulation than other examples. But the important thing is that there's a very, very tiny fraction of TADs that only contain one gene. And most of them, I think it was 5% of TADs contain genes that are all convergently regulated. So even in any scenario, there always is more complicated regulation going on in a TAD. Yeah.
We have a question from the room, Hannah.
Hi, really beautiful talk, thank you. So I was interested in your last point about LAD release, and that LAD release occurs before transcription initiates, and then you said that you saw these attack peaks coming up. Was that before the release? And what do you think the mechanism is? So you think that there's enhancers being activated, and what's the relative timing of those 3 stages?
Yeah, so I wouldn't feel confident to say causal events because these are 2 technologies that sort of measure transcription in slightly different ways. So I don't think we can align them directly and say, within a few hours, this thing is happening before this. I would just say that they occur at the same time. But the only mechanism I can imagine how LAD would get released at specific loci would be through transcription factors that target that specific locus, right? But the LAD release is happening over a period of roughly 24 hours. So I think there's a progressive remodeling in these. Remember, it's a huge domain, right? Like, so the very fact that a relatively small number of tiny regulatory elements can recruit stuff that can remodel everything, I think is surprising. But I also don't know that that's the case, right? So, I think does that answer your question?
May I follow up?
Well, go.
Do you see examples where there's attack peaks only at one end, but still the whole TAD is released?
Yeah, so we don't see that it's not that every region that is releasing from the nuclear envelope, it's always the whole TAD. So we often see in some scenarios that entire TADs can practically remain heterochromatin, and only little regions just turn on and escape this environment. So I think it's more to do with the density of information that you contain. Like if you're a huge domain with lots of enhancers, then maybe you have to, or as a product of that, you release the whole thing. But other domains can completely operate practically in heterochromatin. So it makes predicting, I don't know why that is. And I think that just highlights the general problem with all of this, which is that every locus is slightly different, and so when you're trying to make generalizable statements, particularly when with variant interpretation, it makes it very hard because we just don't understand the rules enough. Yeah.
Very complicated, thank you.
Yeah.
So I actually have one last question about the single cell part. I'm actually quite interested in the technology, but you were quite fast on this. Could you share with us, if you are allowed, if it's like a plate-based method or more 10x kind of method, and what the throughput is? So will we have to do human cell atlas now with the DAM-ID single cell?
I'd be game if you have the money. So it's totally open. It's a plate-based method, so our throughput is relatively low currently. We basically have to express a protein, a methylase that you fuse to a protein of interest, and then this will methylate nearby DNA sequences when the protein is bound, and then this signal accumulates over time. And we then isolate cells and we can process all this stuff. We are trying to improve this so we can try, for example, to have spatially resolved versions of the DAM-T, so you can look in tissue sections at way higher throughput, and that's going to be fun because then it opens up a number of new questions that you can ask, like how does chromatin state vary with position in a tissue?
Good. So since there are no more questions from the audience, I would like to thank everyone for coming, and I would in particular like to thank the speakers. We learned first in the first talk that it's very complicated at one locus, and now we heard from you that it's even much more complicated at any other locus.
It's a satisfying answer.
We have much more to learn for 3D genome architecture, but it's been an incredible session, and I would like you all to thank the speakers once more. Thank you.
Thank you.